M17 compound light microscope. On completion of this module, you should be able to set up and use this microscope or a similar instrument as though by second nature. Some of you will have Nikon microscopes. These differ only slightly from the Vickers, and your demonstrator will explain these differences to you later. The principles behind the preparation and use of both instruments are exactly the same. The controls you will use to set up the microscope are illustrated in this diagram. There are several ways in which light microscopes may be used. Bright field microscopy is the normal mode of operation, on which all other optical modes are based. So you must set up the microscope correctly in this mode before you can use any of the others. When first using a microscope, it is important to ensure that it is properly aligned. If the instrument you wish to use is poorly aligned, then you may correct this using the low power objective only. Once the microscope has been set up correctly using the low power objective, only minor adjustments in alignment and focus should be necessary as you move to the higher power objectives. Turn the microscope on and adjust the voltage control to between 4 and 5 volts. If the light is too bright or too dim, you won't be able to get a good image and your eyes will very quickly become tired. Fully open both the field iris at the base of the microscope and the substage or aperture iris on the underside of the stage. Select the lowest power objective lens, usually 10 times. The magnification is written on the side of each of the objectives. Adjust the interocular distance of the eyepieces to suit your vision. You should see one circular field. A convenient way of setting the focus is to use a plain slide marked with a China graph cross. Place this on the stage of the microscope and focus using the controls on either side of the instrument. Always focus the microscope by looking from the side first. Rack the stage upwards until the slide almost touches the objective lens. Close one eye, look down the left-hand non-adjustable eyepiece and turn the focus controls in the opposite direction so that the slide moves away from the lens. Use the larger coarse focus control first, then the smaller fine focus one to make the image as crisp as possible. Now, using the other eye, look down the adjustable eyepiece and focus the object again by rotating the silver collar. Next, look down the microscope and close the field iris until there is a bright dot in the field of view. Using the control below the stage, raise or lower the substage condenser to bring the field iris into sharp focus. For most microscopes, the iris will be focused when the substage condenser is at, or close to, its uppermost position. If the image of the iris isn't centered in the field of view, use the two opposing finger screws to adjust it. These are located on the sides of the condenser. Now open the field iris until an even field of illumination is just obtained. Remove an eyepiece. Viewing the back focal plane of the objective, adjust the substage iris until approximately 80% of the aperture is illuminated. This eliminates glare and produces a crisp image. Replace the eyepiece. The microscope is now set up correctly for bright field microscopy using the low-power objective. Carefully remove the slide without touching the objective. And replace it with the stained and fixed bacterial specimen. Only minor adjustments with the fine focus control should be required. After examining the specimen under low power, change to the 40 times objective. Quickly check all your settings and make any necessary adjustments. Although not appropriate for today's exercise, you should note that detailed examination of specimens using the 40 times objective should only be undertaken when the specimens are viewed under a cover slip of the correct thickness. To see the bacteria, you'll need to use the 100 times bright field oil immersion objective lens. This is identified by the engraving on the side of the objective. Open the substage or aperture iris fully. Move the 40 times objective to one side and place a small drop of immersion oil onto the area of the slide illuminated by the beam of light. Don't use a cover slip. When you swing the 100 times oil objective into place, a film of oil forms between the slide and the objective. The microscope you are using is parfocal. This means that all the objectives focus at the same point. As a result, you should only need to make small adjustments to the fine focus to obtain a clear image. Move around the slide using the stage controls until you find what you want. In this view, you can see the globular shape of Staphylococcus epidermidis stained with carbofusin. If you cannot focus on the object, the most common explanation is that the slide is upside down. It's possible to focus on an upside down slide using the low power objectives, but not with an oil immersion one. At the end of the day, when you have finished using the microscope, always clean the oil objective thoroughly with lens cleaning tissue. Be careful to avoid contaminating non-oil immersion objectives, the 10 times and 40 times, with oil. To make aseptic transfers, work close to a Bunsen burner turned to the hottest flame setting. This is because the updraft of heated air helps to prevent microorganisms from settling on the apparatus. Degrease the slide by passing it slowly through the Bunsen burner flame. Don't hold it in the flame or it will get too hot. Flame the loop by holding it upright in the hottest part of the Bunsen flame, just above the blue cone, until the whole length of the wire glows red hot. Remove the top from the bottle of distilled water, but do not put it on the bench. You will contaminate it if you do. With practice, you should be able to hold the lid in the crook of your little finger, while holding other objects, such as the loop, in the same hand. Pass the neck of the bottle through the Bunsen burner flame. Remove a loopful of distilled water and pass the neck of the bottle through the flame again. Replace the top on the bottle. Place the loopful of water on the microscope slide. Sterilize your loop again by heating it until it is red hot. Allow the loop to cool in air for about 10 seconds before placing it into the bacterial culture. Remove the top from one of your cultures but do not put it on the bench. Pass the neck of the culture vessel through the Bunsen burner flame before inserting the loop into the sample. It is essential that you pick only a very small quantity of the culture from the surface of the agar. Pass the neck of the vessel through the Bunsen burner flame again and replace the top. Mix the bacterial culture into the drop of water and spread the film thinly. With experience, you will find that you need only a very small amount of material. Allow the slide to air dry. This normally takes a few minutes. Finally, sterilize the loop by carefully heating it to red heat. To avoid splattering live bacteria, the loop should be introduced into the upper, cooler part of the flame so that it heats up gently. It should then be lowered into the central, hotter part of the flame just above the blue cone. With the bacterial film uppermost, fix the bacteria by passing the slide rapidly through the flame several times. Remember to adjust the Bunsen back to a luminous flame when you've finished using it to avoid the danger of setting fire to yourself. The Bunsen burners you are using are fitted with pilot lights. To reduce overheating of the laboratory, please switch to the pilot light when you are not using the full flame. The bacteria are stained using dilute carbofusion. This is a red dye that will stain microbial cells. Place the slide over a sink and flood the fixed film of bacteria with the stain. Leave for about 60 seconds to allow the stain to penetrate the tissue. Then rinse the slide thoroughly under running water. Dry the slide carefully using blotting paper. To ensure that the slide is completely dry, pass it through the Bunsen burner flame, again with the film uppermost. The slide is now ready for examination. Use the lowest power objective, usually 10 times, first. Then move up through the objectives until you are using the 100 times oil immersion objective. You should be able to observe bacteria with this objective. When you have finished with slides, dispose of them in the discard pot provided.
Most microbes, like these found in pond water, are unpigmented. So, if we want to do more than visualize their size, shape and motility, they will have to be stained. A variety of different staining protocols are used by microbiologists to reveal particular microbial structures. In the Gram stain, the bacteria are stained with a crystal violet iodine complex, decolorized with acetone, and are then counterstained with carbylfuchsin. Bacteria which resist decolorization with acetone and retain the crystal violet iodine complex are referred to as gram-positive. In this preparation, the blue-black coccus, Staphylococcus epidermidis, is gram-positive. Bacteria that fail to retain the initial stain are termed gram-negative. The red bacillus, Escherichia coli, is gram-negative. It has lost the crystal violet iodine complex, but has taken up the carbylfuchsin counterstain, making it easier to see. The Gram stain detects a major structural difference between the two groups. Gram-negative bacteria have an extra membrane in their cell envelope, and this is of fundamental importance in bacteriology. Some bacteria, called mycobacteria, will not take up dyes using conventional methods because they have very waxy cell walls. The Zeil-Nielsen staining technique overcomes this using a strong solution of carbylfuchsin. The stain is heated on the slide until the preparation steams. This helps the dye to penetrate the waxy cell walls of the bacteria. After about 10 minutes, the dye is tipped off, and the slide is flooded with a mixture of acid and alcohol. This leaches the dye from all but the acid-alcohol-fast cells. In order to see the structures in the preparation, the background material is usually counterstained with methylene blue. Some mycobacteria are of considerable clinical importance. This is Mycobacterium tuberculosis, which, as the name suggests, causes TB. Mycobacterium leprae causes leprosy. Once stained, mycobacteria can resist decolorization with acids and alcohol, and so are sometimes called acid-alcohol-fast bacilli. Spores may also be difficult to stain, and these too may require heating to be persuaded to take up a dye solution. In this image, spores of a member of the genus Bacillus have been stained with malachite green. The rest of the preparation is visualized using a mercurochrome counterstain. Bacteria may be actively motile. For many bacteria of medical importance, motility is achieved with the aid of whip-like structures called flagella. This image is of a Proteus strain that has been stained so that its flagella can be seen. The bacterium is so motile that a single colony can swarm over the entire surface of an agar plate in just a few hours. In the examples shown previously, stains have been used to highlight structures. There are occasions where the failure of a structure to take up the stain can be used to show its presence. In this preparation, both the bacterial cells and the background have been stained. The halo around each cell, where the stain has not been taken up, is a capsule. Capsules can be important in helping bacteria to cause serious infections. This bacterium is Bacillus anthracis, which causes anthrax. If live bacteria are to be visualized, special microscopic techniques may be employed. Here is a wet preparation of pond water viewed using interference microscopy. Caulobacters are motile bacteria common in aquatic environments, where they clump together to produce rosettes, as seen here. One method of determining bacterial motility is to use a Craigie tube. A hollow tube is placed in semi-solid agar inside a universal bottle. Using aseptic technique, take a sample of the bacterium to be tested. Inoculate the medium inside the hollow tube. Following incubation, examine where bacterial growth has occurred. Non-motile bacteria will remain confined within the tube at the site of inoculation. Motile bacteria will be able to swim out from the bottom of the tube and colonize the surrounding medium. Confirm which bacterium is motile and which is not. Take a sample from outside the hollow tube and, Using aseptic technique, inoculate a nutrient agar plate. Repeat the procedure for the second tube. Streak both plates for single colonies. Following overnight incubation at 37 degrees Celsius, there are no colonies on the first plate, indicating that these bacteria are non-motile. By contrast, colonies have developed on the second plate, so these bacteria were able to escape from the hollow tube into the surrounding medium.
Place a drop of 70% alcohol on a clean microscope slide. Material from cultures of filamentous fungi should be removed using a stiff inoculating wire, not the loop used for manipulations with bacteria or yeasts. Flame the wire by holding it upright in the hottest part of the Bunsen flame, just above the blue cone, until the whole length of the wire glows red hot. You must ensure that the inoculating wire has cooled before placing it in a fungal culture. It should have cooled sufficiently after approximately 10 seconds. Remove the cap from the tube, but do not put it on the bench. Kill any contaminating microorganisms by flaming the neck of the tube. Remove a small amount of the culture. For fungal cultures, it is often useful to take a little of the agar medium together with the fungus. In any case, the material should be disturbed as little as possible when being transferred to the slide. Flame the neck of the tube once more and replace the cap. Immerse the fungal material in the drop of 70% alcohol. This drives out the air trapped between the hyphae. Tease out the material very gently with mounted needles. Do not forget to sterilize the inoculating wire and the needles after use by heating to red heat in a Bunsen flame. Before the alcohol dries out, add 1 or at most 2 drops of lactophenol cotton blue. A common fault is to add too much stain to the preparation. Holding the cover slip between your index finger and thumb, touch one edge of the drop of stain with the edge of the cover slip. Lower the cover slip gently onto the slide, trying to avoid air bubbles. Your preparation is now ready for examination. Make the initial examination using the 10 times low power objective lens. The thinner parts of the preparation, generally around the edges of the mounted material, will yield the best images. In this preparation, we can see sporophores of a Rhizopus species. Switch to the higher power 40 times objective for more detailed examination of spores and other structures. Here we can see the spores that have been released from one of the sporophores we were examining earlier, and the empty sporophore itself. This is a procedure that allows fungal growth to be examined undisturbed. The technique is dependent upon the fungus attaching to a glass cover slip. You are provided with a sterile glass slide on a bent glass support in a Petri dish. Cut a square of agar growth medium that is small enough to fit easily on the glass slide. Carefully transfer this aseptically to the center of the slide. Flame the inoculating wire and allow it to cool before inserting it into the sample. Only a small amount of material is required. Inoculate one edge of the agar square with the fungus to be examined. Repeat the procedure for the other 3 edges. Remove a cover slip from the Petri dish where it has been soaking in alcohol to keep it sterile. Flame the cover slip to burn off the alcohol and place it on top of the agar block. Add a little sterile water to the base of the Petri dish to ensure that the agar does not dry out while the culture is incubating. The duration and temperature of the incubation depend upon the species of fungus being examined. When sufficient growth has occurred, remove the cover slip. Place the cover slip with the fungal growth uppermost onto a clean slide. Add a drop of 70% alcohol to the center of the cover slip followed by a drop of lactophenol cotton blue. Turn the cover slip over and place on a clean slide. When examined with a microscope, the slide culture will frequently enable you to view intact structures. Here we can clearly see chains of Alternaria spores.
Spores and their associated structures are very valuable in the identification of fungal species. As well as helping to disperse the fungus, spores can withstand harsh conditions that could kill other parts of the organism. The spores of Rhizopus oryzae, a simple fungus, are produced asexually in sac-like structures called sporangia. In this preparation, stained with lactophenol cotton blue, the stages in the development of Rhizopus spores are visible. The large pale blue structure is an immature sporangium containing many spores called sporangiospores. As the spores mature, they turn dark brown. Several sporangia containing mature spores can be seen at the top left of this picture. In the centre of this image is a sporangium discharging its spores, and below it, to the right, is one that has already discharged. The dome structure that formed the base of the sporangium, the columella, has collapsed and resembles an umbrella. The sporangia of Rhizopus oryzae are carried on clumps of highly branched hyphae that look like roots. This root-like structure is called a rhizoid. The hyphae of simple fungi, like Rhizopus, are not divided into separate cells by cross walls or septa, and are referred to as being aseptate. This fungus, Penicillium chrysogenum, has no sexual stage, but reproduces entirely through the production of round, asexual spores referred to as conidia. The elongated finger-like cells from which the conidia form are known as phyalides, and these branch from a special hypha known as a conidiophore. The fruiting structure of this fungus resembles a brush. Indeed, the genus gets its name from the Latin word for a small brush. It is fungi of this genus that are used to produce penicillin. The higher fungi have hyphae that are divided into segments with cross walls known as septa. To ensure that all the cells or compartments in a hypha carry a nucleus, in fungi such as Coriolus versicolor, the junctions between cells are marked by special structures known as clamp connections. These are D-shaped features that you can see on the sides of the hyphae. Most fungi can be classified as molds or yeasts. Molds, like Rhizopus, produce thread-like structures or hyphae. Yeasts, like Saccharomyces cerevisiae, do not. This is the yeast that is used in baking and brewing. It has oval cells and, like the majority of yeasts, it reproduces by a process of budding. As well as budding, Saccharomyces cerevisiae can also engage in sexual reproduction. This leads to the formation of several asci, each of which contains 2 or 4 ascospores in this species. In this preparation, stained using a modification of the Zeil-Nielsen technique, the ascospores appear red. The ascus produced by Saccharomyces cerevisiae is a relatively simple structure. Other fungi in this family produce complex fruiting bodies called ascocarps. The ascocarp of Sordaria formicola is a large, flask-shaped structure known as a perithecium. You can see asci being expelled from this perithecium through an ostiole, a narrow opening in the top. Each ascus contains 8 ascospores. Immature ascospores readily take up the blue lactophenol stain. As they mature, they become darker. These asci contain ascospores at different degrees of maturity. Not all yeasts reproduce by budding. Cells of Schizosaccharomyces pombe elongate until they reach a critical size, and then divide into 2 by a process of binary fission. Several of the cells in this field are about to split into 2, a cross wall separating the parental cells into 2 offspring. Bacteria also reproduce in this way. Some fungi do not reproduce sexually, but can form elaborate asexual sporing bodies. The numerous dark pear-shaped structures in this image are conidia, asexual spores of the genus Stemphylium. They are divided into separate cells by septa that may run across or up and down the conidium. Zygorhynchus muelleri produces spores sexually and asexually. The spores formed through sexual reproduction are called zygospores and appear as large, brown, warty structures. Some fungi need strains of different types to mate. In Zygorhynchus muelleri, 2 hyphae of the same strain may come together to reproduce sexually. Zygospores are generated between the tips of 2 hyphae that form structures called suspensors. These are of different sizes. The larger suspensor is clearly visible in this preparation, but, as is typical, the smaller one cannot be seen. The fungus Emericella nidulans, also called Aspergillus nidulans, reproduces both sexually and asexually. The large, dark, spherical structure is a cleistothecium, an ascocarp produced by sexual reproduction. The ascocarp is surrounded by a mass of HeLa cells. The function of these colourless, thick-walled cells is not known. When the cleistothecium breaks, numerous small, round asci, each containing 8 red ascospores, are released. The ascospores of this species have 2 equatorial crests, making them appear like a pulley wheel, but these are only clearly visible under electron microscopy.
To make aseptic transfers, work close to a Bunsen burner turned to the hottest flame setting. Place a rubber teat on the end of a sterile Pasteur pipette. Be sure to do this in a way that prevents glass digging into your hand should the pipette break. Remove the cap from a tube containing sterile nutrient broth. Flame the neck of the tube and then withdraw a small volume of broth using the Pasteur pipette. Flame the neck of the tube again and replace its cap. Open the empty sterile tube and flame its neck. Place the broth from the Pasteur pipette into the empty tube. Flame the neck of the tube again and replace its cap. Dispose of the Pasteur pipette in the discard jar. Label both tubes with your initials and bench number and place them in the rack provided. they will be incubated at 37 degrees centigrade. Aseptic technique achieves two things. First, it protects you from your cultures. Second, it protects your cultures from you and other sources of contamination in the environment. You are provided with a suspension of two bacterial species. The aim is to inoculate a plate of nutrient agar so that, following incubation, separate colonies of each microorganism may be readily distinguished and isolated as pure cultures. As demonstrated here with felt-tip pens, the principle is to draw out ever-decreasing amounts of material across the surface of the agar. By the end of the process, maximal dilution of the microorganisms present in the original suspension is achieved. During this procedure, it is important to use as much of the surface area of the agar as possible. When you first examine the mixed culture of bacteria, you may find that the cells have formed a pellet at the bottom of the tube. It is essential that you resuspend the bacteria by gentle agitation of the culture. However, be careful not to create an aerosol, as this is a highly efficient way to disperse microorganisms. Flame the loop by holding it upright in the hottest part of the Bunsen flame, just above the blue cone, until the whole length of the wire glows red hot. You must ensure that the loop has cooled before placing it in a bacterial culture. It should have cooled sufficiently after approximately 10 seconds in air. Remove the cap from the tube, but do not put it on the bench. Flame the neck of the tube to kill any contaminating microorganisms. Place the loop into the suspension of bacteria, then withdraw it carefully. Flame the neck of the tube once more and replace the cap. Pick up a nutrient agar plate and hold it with the agar surface upwards near to the convection currents of a Bunsen burner turned to the hottest flame setting. The updraft of heated air prevents microorganisms from settling on the agar. Inoculate a small area of the plate with a smear of the bacterial suspension using a circular motion. You should brush the surface gently with the loop, taking care that it does not dig into the nutrient agar. Next, sterilize the loop by carefully heating it to red heat. By sterilizing the loop, you ensure that you don't carry over too many organisms from the original inoculum. Cool the loop by plunging it into an area of agar where there are no microorganisms. Then make 3 or 4 streaks along one side of the plate, drawing out material from the original inoculum. Sterilize the loop, cool it, and, at an angle to the first set of streaks, make another 3 or 4 streaks along the side of the plate. Repeat the procedure for a third time. Finally, sterilize the loop, cool it, and make one continuous zigzag streak across the plate without touching any of the areas you've already streaked. Replace the plate upside down in its lid. This prevents condensation dripping onto the culture during incubation. On completion of inoculation procedures, it is important to sterilize the loop. Label the base of the plate with your initials and bench letter. After the appropriate incubation period, if you've streaked the plate correctly, you will have well-separated colonies which you can identify and use for further experiments. This plate was inoculated with a mixed culture of Escherichia coli and Staphylococcus epidermidis. E. coli forms large colonies, while the Staphylococcus epidermidis colonies are very small. Flame and cool the loop, then pick off a single colony of either E. coli or Staphylococcus epidermidis. Using the crook of your little finger, remove the cap from a tube containing sterile nutrient broth. Flame the neck of the tube, then introduce the inoculum on the loop. Holding the tube at an angle, rub the inoculum on the glass so that, when held upright, the inoculum will be below the surface of the broth. This procedure avoids wetting the loop excessively and thus reduces the risk of generating aerosols which lead to contamination. Flame the neck of the tube and replace the cap. Sterilize the loop. Label the tube with your initials, bench letter, and the name of the bacterium being cultured. The Gram stain, developed in 1884 by the Danish bacteriologist Hans Christian Gram, divides bacteria into two large groups, gram-positive and gram-negative. Bacteria are stained with a violet dye iodine complex, decolorized with acetone, and counterstained with the red dye carbofusin. Prepare a heat-fixed film of the specimen. Flood the film with oxalate crystal violet solution for about 30 seconds. Wash the slide with plenty of running water. Drain the excess water and flood with fresh Lugol's iodine. Leave for 1 minute. The next step is the only critical step in the gram staining process. Care must be taken to obtain the correct degree of decolorization. The film is decolorized with acetone. First, wash the iodine off the slide with running water. Drain the excess water and flood the slide with acetone. Wash the acetone off immediately using plenty of running water. Drain the excess water and flood the film with dilute carbofusin. After about 30 seconds, wash the carbofusin off under running water. Blot the film dry and remove any residual moisture by heating gently in a Bunsen flame. The film is now ready for microscopic examination using the 100 times oil immersion objective. In this preparation, the gram-positive coccus, Staphylococcus epidermidis, has retained the initial stain and appears violet, while the gram-negative rod, Escherichia coli, has lost the initial stain complex but has taken up the counter stain and appears red. These staining characteristics are due to major structural differences between the cell walls of the two groups.
In the early days of bacteriology, broths, made by boiling meat, were used as growth media, with gelatin used as the solidifying agent. If solid media were required, sliced vegetables could be used. Reddish colour in this slice of potato is due to the presence of Serratia marcescens, a bacterium commonly found in the environment. Agar quickly replaced gelatin as the gelling agent, and it is yet to be bettered as the solid support for bacteriological cultures. These days, we can buy a range of growth media, all of which have been subject to careful quality control. Simple defined media may be made by mixing the component parts, but it is generally much more convenient to use commercially supplied media. Making a growth medium is quite a simple process. Measure out the appropriate weight of powder and the relevant volume of distilled water, and make sure you wash all the powder into the mixture. If the medium contains agar, the mixture must also be heated to ensure that the agar melts and is evenly distributed. The medium is sterilized by autoclave treatment. Heat labile components must be sterilized by other means. This defibrinated horse blood was withdrawn aseptically, so it is already sterile. Prepare fresh blood agar plates by adding the blood to cool molten agar. Remember, agar does not re-solidify until about 45 degrees Celsius, so you can add the blood to the sterile medium once it has cooled sufficiently. Mix the blood with the agar by gently inverting the bottle several times. If you add fresh blood to hot agar, it undergoes a change to give a rich chocolate appearance to the medium. Pour a little of the molten agar into each plate. Make sure that the medium is evenly distributed and lay the plates out on the bench to set. Once the agar has set, invert the plates before you store them. This prevents any condensation falling from the lids onto the agar. If a large number of plates are required, plate pouring machines can be used. You may need to use agar slopes rather than plates. To make these, pour the molten agar into tubes prior to autoclaving. Following sterilisation, place the tubes at an angle while the agar is still liquid and leave them to set in this position. Inevitably, pouring warm liquid into a Petri dish will cause condensation. Before plating a culture, the surface of the agar is dried. Plates are stored open and inverted in a warm atmosphere until all the surface water has evaporated. They are then ready for inoculation. Following incubation, bacteria can be seen as colonies growing on the surface of the medium. Heat can be used to kill microbes, and different organisms have different susceptibilities to heat. Exposing bacteria to a range of temperatures and then subculturing these cultures will show if bacteria have survived or not. This is how to determine the thermal death point of a culture. You are provided with 3 cultures and 4 sterile tubes for each. In addition, 1 sample for each culture has already been autoclaved for 15 minutes. This tube is marked with tape showing dark brown stripes, indicating a successful autoclave treatment. Autoclave tape appears uniformly buff until it has been exposed to the appropriate temperature in the autoclave, when brown stripes develop. Using aseptic technique, dispense a small volume of the first untreated sample into its 4 sterile tubes. Repeat the procedure for all 3 bacterial cultures. Remember to use a new pipette for each sample. Three of the 4 sets of tubes should be placed in preset water baths and exposed to temperatures of 55, 65 and 75 degrees Celsius. To expose cultures to a temperature of 100 degrees Celsius, use a boiling water bath. Place some water in the bottom of an enamel mug and bring it to the boil on a tripod placed over a Bunsen burner. Do not place much water in the mug, since it will take too long to reach boiling point. Once it is boiling, carefully place your test cultures in the mug. Tubes should be left in the boiling water or in the relevant water bath for 15 minutes. Having exposed your cultures to a range of temperatures, you should now test for survival of bacteria within the culture by placing them onto nutrient agar. For each test organism, divide a nutrient agar plate into 6 sections. Label each section C for the control culture, followed by the 5 different temperatures to which the cultures have been exposed. The first section is inoculated with the original culture, which has not been exposed to elevated temperatures. Each of the remaining cultures will be inoculated onto the relevant section of the plate. Remember, this is an aseptic procedure. Flame the loop between samples. Do not put the cap on the bench, and flame the top of the tube before and after sampling. Repeat the procedure for all 3 cultures. The plates are incubated at 37 degrees Celsius overnight. The bacteria on this plate indicate survival of the unexposed control and of bacteria exposed to 55 and 65 degrees, but not to higher temperature. This indicates that the thermal death point for this organism is between 65 and 75 degrees Celsius. On this plate, the bacterium has survived exposure to elevated temperatures less well. There is only growth of the control bacterium and of cells exposed to 55 degrees Celsius. This bacterium, therefore, has a thermal death point that lies between 55 and 65 degrees Celsius. This plate shows some growth in all divisions except that inoculated with the autoclaved culture. there is a luxuriant growth of bacteria exposed to temperatures of 55 and 65 degrees Celsius. At 75, there is a slight drop in the number of colonies. This is because vegetative cells in the culture are susceptible to exposure at this temperature. Even after boiling for 15 minutes, a few colonies can be seen. These result from the germination of heat-resistant spores that have survived the boiling. Exposure to moist heat in an autoclave will kill even these spores. You have not been told which culture is which. You should work this out by examining a gram film prepared from the bacteria from each set of cultures. Remember, E. coli is a rod-shaped bacterium that is gram-negative. It cannot retain the crystal violet iodine complex of the gram stain, and so it appears as small red rods when viewed under the microscope. Both Enterococcus faecalis and Bacillus subtilis are gram-positive and retain the crystal violet iodine stain complex. Their shape can differentiate them. Members of the genus Bacillus, as their name suggests, are rod-shaped. Some cells contain spores, and these can be seen as an absence of staining. Enterococci have round or roundish cells. When working with microbes, it is very important to make sure that they can be disposed of in a safe manner. Small, disposable items are often placed in a discard jar once they've been used. But how do we know that the disinfectant is doing its job properly? One way in which we can test this is with the in-use test. The first step in the in-use test is to remove a measured sample of the disinfectant from the discard jar. Use the large pipetta for this. We need to count the number of microbes that have survived in this sample of disinfectant. Using aseptic technique, 1 ml of disinfectant is diluted with 9 ml of a solution of lecithin. The disinfectant is diluted to ensure that it doesn't continue to act once the sample has been taken. Lecithin will inactivate the disinfectant used in our laboratories. You now have a 1 in 10 dilution of your disinfectant. To find out if there are any viable microbes present in your sample, you will need 2 well-dried nutrient agar plates. If you are working with fastidious organisms, which are difficult to grow, you may need to use a rich growth medium for this test. For example, fresh or heated blood agar. In general, however, this is not necessary. Using the smaller pipetta and aseptic technique, dispense 0.2 ml of diluted disinfectant onto each plate. Spread the inoculum evenly over the surface of the plate with a sterile bent glass rod provided. Label the plates and incubate one overnight at 37 degrees, leaving the other at room temperature. Following incubation, examine both plates for bacterial growth. Remember that the in-use test is performed to ensure that the disinfectant in your jar is working properly. If it is, it will kill any microbes that may be present. If the test is satisfactory, then you should see no growth on either plate. Because disinfectants may not kill all microbes immediately on contact, you may observe up to 5 colonies on a plate in the in-use test. Both these plates would still be regarded as satisfactory. Bacteria forming the colonies that grow on the plate on the right, which was incubated at 37 degrees, have evolved to grow at that temperature. Such bacteria are often of human origin, 37 degrees Celsius being body temperature. They may have come from your microflora, and some of these bacteria, if they get in the wrong place, can cause serious infections. Colonies growing on this plate include both skin and gut organisms. Disinfectant yielding such a result has failed the in-use test and will require urgent further investigation. Bacteria growing on the plate at room temperature are most likely those found in the laboratory environment. Although the colony on this plate is very large, it has grown from a single spore of Bacillus cereus var. Mycoides. Since there is only one colony here, the test in this case is satisfactory, and the disinfectant can be regarded as safe. The bacteria on this nutrient agar plate have not been able to grow anywhere near the white discs. These contain antibiotics, the discovery of which was one of the greatest triumphs of 20th century medicine. During the Second World War, these drugs were more valuable than gold, but afterwards they soon became available to the public. On this plate, the fungus in the centre is producing a large quantity of antibiotic, which has inhibited the growth of the sensitive bacteria spread as a lawn over the whole plate. This was the basis of Alexander Fleming's observation. Bacterial susceptibility to antibiotics can also be tested using a controlled sensitivity test. The susceptibility of the test culture is compared with a control culture of bacteria known to be sensitive to the antibiotic to be tested. Both cultures are diluted to approximately 10 to the 5 colony-forming units per mil. This concentration of cells will give a semi-confluent growth. If there are too many cells inoculated, there is a risk that some of the sensitive bacteria will appear resistant because of the size of the inoculum. This is the so-called inoculum effect. If there are too few bacteria, the zones of inhibition will not be seen clearly. The test plate is divided into 3 sections. Control strain is inoculated in the centre section. The cotton bud fell onto the autoclave tinfoil lid of the container, not onto the bench, so it is still sterile and in this case can be used to inoculate the sample. If the swab touches the bench, do not use it. It will no longer be sterile. The test strain is inoculated on both of the outer sections of the plate. After inoculating the plate with both the test and control strains, the next step is to apply the antibiotic discs to the culture. Four discs, each containing a different antibiotic, are carefully placed at the interface between the test and the control inocular. The plates are then incubated overnight. Following incubation, the growth of the test and control bacteria can be observed. If the test has worked successfully, the control strain will fail to grow in the area close to each disc. This is because active antibiotic has diffused out from each disc and has either killed the control bacterium or has inhibited its growth. If the test strain is resistant to the antibiotic, it will grow happily in its presence, and no zone of clearing in the bacterial lawn will be seen near the disc. It is important to spread the inocular evenly over the surface of the different sections of the plate. It is also important to inoculate the control organism before inoculating the test strain. This prevents accidental contamination of the sensitive control strain by resistant bacteria in the test strain. Take care not to inoculate the test strain over the control. Here, the resistant test strain in the upper section of the plate has been inoculated over the control strain, and inhibition of the control strain has been obscured. This makes interpretation of the test difficult. The importance of this test for antimicrobial sensitivity is that it is a controlled sensitivity test. You can be assured that if the test bacterium grows up to an antibiotic disc, that strain is resistant to that antimicrobial agent. This is because antibiotic from the disc will have inhibited the control strain. If the control strain also grows up to the disc, this shows that the disc didn't contain active antibiotic for some reason. In such cases, the susceptibility test needs to be repeated for the antibiotic that didn't work properly. With particular antibiotics, careful examination of the zone of inhibition will show a few isolated colonies growing where most of the bacteria have been inhibited. These are spontaneous mutants that have become resistant to the antibiotic being tested. In laboratories where large numbers of sensitivity tests are performed, a rotary plater may be used. The uninoculated plate is placed on a turntable and is rotated. The control strain is inoculated in the center of the plate. The swab is dragged from the center of the plate to the silver ring, visible on the turntable. The test strain is then inoculated on the remainder of the plate, moving the swab from the outer edge of the plate towards the silver ring. Having inoculated the plate with the rotary plater, antibiotic discs must be placed at the interface between the test and control inocular. This can be facilitated using an automatic disc dispenser. These hold up to 8 cassettes, each containing up to 50 discs impregnated with antibiotics. This means that different antibiotic combinations can be used to test the susceptibility of a range of bacteria. The dispenser automatically dispenses discs onto the inoculated plate when the plunger is depressed. The ring of discs coincides with the silver ring on the rotary plater's turntable. The test on the left shows a strain that is fully sensitive to all of the antibiotics being tested. The plate on the right highlights a possible problem. If antibiotic discs have not been stored properly, they will lose their potency. The control bacterium has only been inhibited by 2 of the antibiotics tested on this plate. Consequently, the susceptibility of the test strain to these antibiotics must be retested. Dry heat is less effective in killing microbes than is moist heat. To test this, 4 universal bottles have been prepared with a dry application of spores of Bacillus subtilis. The cap of the first bottle is screwed down tight. This will prevent steam from within the autoclave entering the bottle. A couple of drops of sterile distilled water are added to the second bottle, and its cap is also screwed down tight. This water will generate steam within the bottle during the autoclave process. The caps of the third and fourth bottles are also screwed down. The first 2 bottles are subjected to an autoclave run. Autoclaves are simply giant pressure cookers that kill microbes by exposing them to steam under pressure. In this case, the contents of the autoclave are held at 115 degrees Celsius for 15 minutes. The third bottle is placed in a hot air oven and held at 160 degrees Celsius for 2 hours. The first hour allows the contents of the oven to reach the desired temperature for sterilisation, and the second hour is for the sterilisation process itself. The fourth bottle, which is not exposed to elevated temperatures, acts as a control to demonstrate the viability of the bacterial spores throughout the experiment. Using aseptic technique, a small volume of broth is added to each of the universal bottles. They are incubated overnight at 37 degrees Celsius. Following incubation, the bottles are inspected for growth. The control is turbid, indicating the viability of spores throughout the experiment. The bottle that had its cap screwed down tightly prior to autoclaving has a cloudy broth, indicating bacterial growth and hence failure of the autoclave process. The spores that were held at 115 degrees Celsius in steam have failed to survive, as shown by the clear appearance of the broth. The culture exposed to dry heat is also clear, showing that spores have not survived dry heat either. Remember, the spores that were present in the control bottle, which was not heat-treated, have survived and have germinated on culture. This gives the broth in the final bottle its cloudy appearance. Bacterial cells, like those in this suspension, can be killed by exposure to ultraviolet light. To demonstrate this, a bacterial lawn is made by flooding the surface of a well-dried nutrient agar plate with a test suspension. The liquid is drained off. Any excess is removed from the surface of the agar with a pipette. After allowing the lawn to dry, the plate is everted and the base divided into strips. It is then inverted again and shielded with card before being exposed to an ultraviolet light source section by section. The last part of the plate is left unexposed to demonstrate the viability of the test bacteria. The plates are incubated to allow any viable cells remaining in the lawn to grow. They were everted prior to incubation, so the unexposed section is on the left. Different bacteria have different susceptibilities to ultraviolet light. Each strain tested will yield the occasional UV light-resistant mutant. Bacillus subtilis can survive particularly well because it produces spores resistant to UV light. This effect is extremely localized. An alternative method of sterilizing a contaminated sample is to filter it. The sample is passed through a membrane filter that is fine enough to trap bacterial cells. A filter is placed in a special holder. Following sterilization in an autoclave, the filter assembly is attached to a 20 ml syringe and placed onto a sterile universal bottle. The syringe is used to force the contaminated liquid through the filter. The filtrate is tested for sterility by plating onto a nutrient agar plate. The original sample was contaminated with bacteria. After filtration, the culture is sterile. To prove conclusively that bacteria were trapped by the filter, it is placed on an agar plate. Bacterial growth is clearly visible following incubation. Different designs of filter are available for different volumes of fluid. We can get a rough idea of the antibiotics to which a bacterium is resistant by performing a disc diffusion test of antibacterial susceptibility. To get a more accurate measure of resistance, we need to estimate the MIC, minimum inhibitory concentration of the antibiotic. First step is to make a series of doubling dilutions of the drug into broth culture. Making the serial dilutions directly from the stock solution would result in an accumulation of petting errors. To avoid this, the dilutions are made into distilled water and then transferred into the broth. Different volumes of the antibiotic stock solution are mixed with various volumes of diluent. This yields a range of concentrations to be added to the broth. The antibiotic stock solution is then diluted further into a second universal bottle. This is used to make up the next 4 dilutions. We now have all the doubling dilutions of the antibiotic ready to add to the nutrient broth. Starting with the most dilute, a sample from each suspension is added to the appropriate tube. The most concentrated of the dilution series, representing 256 milligrams per litre, is produced by inoculating the broth with the original stock solution of antibiotic. A control tube containing broth but no antibiotic is used to check the viability of the bacterium being tested. Next, a colony of the bacterium is inoculated into antibiotic-free broth. This light suspension will be used to inoculate the test broths. Starting with the most dilute, 1 drop of the bacterial suspension is added to each tube in the series. The tubes are incubated overnight at 37 degrees Celsius. Growth of the inoculum is indicated by turbidity in the broth. The antibiotic-free control is cloudy, indicating that the bacteria are viable. Bacteria can grow at low concentrations of the antibiotic. The minimum inhibitory concentration is the lowest concentration that prevents bacterial growth, resulting in a clear broth. In this example, the MIC is 16 milligrams per litre. This test does not distinguish between antibiotics that kill bacteria, the cidal drugs, and those that merely inhibit their growth, static drugs. To establish whether an antibiotic is bactericidal or bacteriostatic, we need to compare its minimum inhibitory concentration with its minimum bactericidal concentration. We are not interested in the dilutions that failed to prevent bacterial growth, so all the cloudy tubes can be discarded. Each of the tubes that show no bacterial growth is then sampled. The samples are plated out for single colonies onto medium that does not contain the antibiotic. The plates are incubated to give any viable bacteria time to produce colonies and are then examined for growth. If the antibiotic is cidal, the bacteria will have been killed by exposure to the drug, and the subcultured plates will show no bacterial growth, as here. If a drug is bactericidal, therefore, the minimum bactericidal concentration and the minimum inhibitory concentration will be the same. If, on the other hand, the drug is static and only inhibits bacterial growth, Removing the cells to the fresh medium without antibiotic present will permit growth to resume. Following incubation, colonies will develop on some of the plates. The minimum bactericidal concentration, the lowest dilution at which no growth is observed following subculture onto antibiotic-free medium, is 256 milligrams per litre in this example. The minimum inhibitory concentration was 32 milligrams per litre, so this antibiotic is bacteriostatic. When determining the thermal death point of bacteria, cultures are examined following incubation. Bacteria on this plate indicate survival of the unexposed control and of bacteria exposed to 55 and 65 degrees, but not to higher temperature. This indicates that the thermal death point for this organism is between 65 and 75 degrees Celsius. On this plate, the bacterium has survived exposure to elevated temperatures less well. There is only growth of the control bacterium and of cells exposed to 55 degrees Celsius. This bacterium, therefore, has a thermal death point that lies between 55 and 65 degrees Celsius. This plate shows some growth in all divisions except that inoculated with the autoclaved culture. There is a luxuriant growth of bacteria exposed to temperatures of 55 and 65 degrees Celsius. At 75, there is a slight drop in the number of colonies. This is because vegetative cells in the culture are susceptible to exposure at this temperature. Even after boiling for 15 minutes, a few colonies can be seen. These result from the germination of heat-resistant spores that have survived the boiling. Exposure to moist heat in an autoclave will kill even these spores. You have not been told which culture is which. You should work this out by examining a gram film prepared from the bacteria from each set of cultures. Remember, E. coli is a rod-shaped bacterium that is gram-negative. It cannot retain the crystal violet iodine complex of the gram stain, and so it appears as small red rods when viewed under the microscope. Both Enterococcus faecalis and Bacillus subtilis are gram-positive and retain the crystal violet iodine stain complex. Their shape can differentiate them. Members of the genus Bacillus, as their name suggests, are rod-shaped. Some cells contain spores, and these can be seen as an absence of staining. Enterococci have round or roundish cells. Following incubation of the in-use tests, examine both plates for bacterial growth. Remember that the in-use test is performed to ensure that the disinfectant in your jar is working properly. If it is, it will kill any microbes that may be present. If the test is satisfactory, then you should see no growth on either plate. Because disinfectants may not kill all microbes immediately on contact, you may observe up to 5 colonies on a plate in the in-use test. Both these plates would still be regarded as satisfactory. Bacteria forming the colonies that grow on the plate on the right, which was incubated at 37 degrees, have evolved to grow at that temperature. Such bacteria are often of human origin, 37 degrees Celsius being body temperature. They may have come from your microflora, and some of these bacteria, if they get in the wrong place, can cause serious infections. Colonies growing on this plate include both skin and gut organisms. Disinfectant yielding such a result has failed the in-use test and will require urgent further investigation. Bacteria growing on the plate at room temperature are, most likely, those found in the laboratory environment. Although the colony on this plate is very large, it has grown from a single spore of Bacillus cereus var. Mycoides. Since there is only one colony here, the test in this case is satisfactory, and the disinfectant can be regarded as safe. Following incubation of the antibiotic sensitivity test plates, the growth of the test and control bacteria can be observed. If the test has worked successfully, the control strain will fail to grow in the area close to each disc. This is because active antibiotic has diffused out from each disc and has either killed the control bacterium or has inhibited its growth. If the test strain is resistant to the antibiotic, it will grow happily in its presence, and no zone of clearing in the bacterial lawn will be seen near the disc. It is hard to imagine life without oxygen, but it can be very toxic. Obligate anaerobic bacteria have not evolved defences against the toxic effects of oxygen and must be protected from it. In the laboratory, we can maintain cultures of obligate anaerobes by growing them in a broth containing minced meat. Textured vegetable protein is preferred these days. The reducing agents in the proteins mop up residual traces of oxygen, permitting growth of anaerobic bacteria. Some species of obligate anaerobe use the protein as an energy source, digesting the minced meat and leaving a rather smelly sludge at the bottom of the broth. Others use sugars present in the broth as their source of energy, and these leave the meat particles intact. In this experiment, you will test the ability of 3 different bacteria, E. coli, Pseudomonas aeruginosa and Clostridium sporogenes, to grow in the absence or presence of oxygen. Take a plate and inoculate it with a culture of Clostridium sporogenes, spreading your inoculum for single colonies. Note that the lid of the Petri dish has 3 small plastic notches in it. These separate the base of the plate from the lid and allow free exchange of gases over the surface of the culture medium during incubation. Between streaks, return your Petri dish to its lid. Remember to cool your loop in the uninoculated agar before plating out. And also remember to streak out only from the previous streak. Take a second plate and inoculate half with E. coli and half with Pseudomonas aeruginosa. first 2 plates will be incubated in an anaerobic jar. The lid has a catalyst in a wire mesh container. A gas pack anaerobic generator will be used to generate hydrogen. The gas pack is activated by the addition of 10 mL of distilled water. The hydrogen produced from the gas pack will then react with oxygen in the jar on the surface of the catalyst, producing water. A recent development in anaerobic bacteriology has been the introduction of generating systems that do not require the addition of water. An airtight box is filled with culture plates ready for incubation. 2 anaerobic atmosphere generator packs are then removed from their packaging and placed in the box in the compartments provided. The lid is clipped into place and the box is ready for incubation at the chosen temperature. Laboratories that process large quantities of plates may use an anaerobic workstation. A third plate is divided into 3 sections and inoculated with all 3 bacteria. There is not enough room to streak these cultures for single colonies. This plate will be incubated in air. All 3 plates are incubated at 37 degrees Celsius. Following incubation, it should be clear which cultures are anaerobic and which are not. This plate, inoculated with Clostridium sporogenes, was incubated in the anaerobic jar. Clostridium sporogenes is an obligate anaerobe and will only grow if oxygen is absent. This plate was inoculated with all 3 cultures and incubated in air. The inoculum of Clostridium sporogenes has not grown, since this is an obligate anaerobe. The culture of Pseudomonas aeruginosa, the obligate aerobe, has grown, as has the facultatively anaerobic E. coli. This plate, inoculated with E. coli and Pseudomonas aeruginosa, was incubated anaerobically. The E. coli has grown, but the Pseudomonas aeruginosa has not, indicating that it is an obligate aerobe. E. coli can grow in the absence as well as in the presence of oxygen. This is why it is described as a facultative anaerobe. This plate was also inoculated with E. coli and Pseudomonas aeruginosa. and was also incubated anaerobically. In spite of this, weak growth of Pseudomonas aeruginosa, an obligate aerobe, can be observed. On rich medium, this bacterium can sometimes exploit nitrogen compounds rather than oxygen in its respiratory metabolism. An alternative method of testing for the effects of atmosphere on bacterial growth is to exploit the stab culture. Label 3 tubes of semi-solid agar with the names of the 3 bacteria being studied. Inoculate a bacteriological loop with Clostridium sporogenes. Using aseptic technique, plunge the loop into one of the tubes. Oxygen will only penetrate a little way into the agar. Repeat this procedure to inoculate tubes with E. coli and Pseudomonas aeruginosa. Following overnight incubation in air at 37 degrees Celsius, prepare your cultures with a control tube on the right containing uninoculated agar. From the left, Clostridium sporogenes, an obligate anaerobe, can grow in the depths of the agar but will not grow near the surface. Next to it, E. coli, a facultative anaerobe, can grow along the whole length of the stab. In contrast, Pseudomonas aeruginosa, an obligate aerobe, can only grow at the surface of the medium. Successful bacterial growth in artificial culture is not only dependent upon providing the appropriate atmosphere, it is also important to provide a suitable temperature for the cultivation of bacteria. Label each of 6 plates with the names of the bacteria being studied. Using aseptic technique, inoculate the first section with a culture of Flavobacterium. Inoculate the next 3 sections with E. coli, Bacillus styrothermophilus and Staphylococcus aureus. Inoculate the other 5 plates in the same way. Following overnight incubation at the appropriate temperature, examine the plates for bacterial growth. Only Flavobacteria grow at low temperature. These bacteria can grow, albeit slowly, at temperatures as low as 5 degrees Celsius, and they can be important spoilage organisms of refrigerated food. All the organisms except Bacillus styrothermophilus grow on the plate which was incubated at 18 degrees Celsius. Both E. coli and Staphylococcus aureus still grow at 30 degrees Celsius. These bacteria have evolved to live in association with humans and can grow very happily at body temperature, 37 degrees Celsius. E. coli can still survive at 45 degrees Celsius, a temperature that kills Staphylococcus aureus. As its name suggests, Bacillus styrothermophilus only grows at elevated temperatures. This plate was incubated at 56 degrees Celsius. Following incubation, it should be clear which bacteria are anaerobic and which are not. This plate, inoculated with Clostridium sporogenes, was incubated in the anaerobic jar. Clostridium sporogenes is an obligate anaerobe and will only grow if oxygen is absent. This plate was inoculated with all three cultures and incubated in air. The inoculum of Clostridium sporogenes has not grown, since this is an obligate anaerobe. The culture of Pseudomonas aeruginosa, the obligate aerobe, has grown, as has the facultatively anaerobic E. coli. This plate, inoculated with E. coli and Pseudomonas aeruginosa, was incubated anaerobically. The E. coli has grown, but the Pseudomonas aeruginosa has not, indicating that it is an obligate aerobe. E. coli can grow in the absence as well as in the presence of oxygen. This is why it is described as a facultative anaerobe. This plate was also inoculated with E. coli and Pseudomonas aeruginosa, and was also incubated anaerobically. In spite of this, weak growth of Pseudomonas aeruginosa, an obligate aerobe, can be observed. On rich medium, this bacterium can sometimes exploit nitrogen compounds rather than oxygen in its respiratory metabolism. Following overnight incubation of stab cultures grown in air at 37 degrees Celsius, compare your cultures with a control tube on the right containing uninoculated agar. From the left, Clostridium sporogenes, an obligate anaerobe, can grow in the depths of the agar but will not grow near the surface. Next to it, E. coli, a facultative anaerobe, can grow along the whole length of the stab. In contrast, Pseudomonas aeruginosa, an obligate aerobe, can only grow at the surface of the medium. When determining the effect of temperature on bacterial growth, following overnight incubation at the appropriate temperature, examine the plates for bacterial growth. Only flavour bacteria grow at low temperature. These bacteria can grow, albeit slowly, at temperatures as low as 5 degrees Celsius, and they can be important spoilage organisms of refrigerated food. All the organisms except Bacillus styrothermophilus grow on the plate which was incubated at 18 degrees Celsius. Both E. coli and Staphylococcus aureus still grow at 30 degrees Celsius. These bacteria have evolved to live in association with humans and can grow very happily at body temperature, 37 degrees Celsius. E. coli can still survive at 45 degrees Celsius, a temperature that kills Staphylococcus aureus. As its name suggests, Bacillus styrothermophilus only grows at elevated temperatures. This plate was incubated at 56 degrees Celsius.